shall I start? I think I shall start. Okay, so I don't know if it's a privilege to have the last talk or not. I got a feeling it's not, but still. Anyway, I'll try and be sort of a little bit more general and try and bring back a couple of the concepts that have come up in the course. My interest has been really in the structural analysis of the over the years, the structural analysis of the opiate system, and like Charlie, I've sort of grown up with it, and so I do know a lot about the opiate system, but not about much else. I don't think so. Anyway, so I was going to tell you how things have sort of evolved to where we are now and what we think of the receptors, because I think it's kind of important that you guys get concepts of how diverse these receptors really are, and how ligands can individually signal through these receptors. I mean, Charlie showed you a very, very nice example with the kappa antagonists, JD-TIC and was it Norby and I, and showed that, you know, although they're antagonists, they're signaling, and they're signaling in such a way which produces, you know, somehow inactivates the receptor. I know Charlie's trying to figure out how that works, but you know, these receptors respond differently to almost every drug you chuck on them. Like it?
Yeah, I love it.
You know, I like making these silly things. So anyway, I guess this doesn't work, does it? Oh, maybe it does. I don't know. Which one? Oh, this was the laser pointer, right? So, you know, I mean, obviously, I don't want to belabor all the stuff because you've already gone through this. You know, there's lots of reasons why people are going to take drugs in the first place. You know, their peers, impulsivity, mood, stress, and obviously this becomes the first site where drugs can help. You know, drugs can help change the endophenotypes, which make you susceptible to addiction. So that's the first site we could look at. When you take the drug, again, this is a site where you can maybe modulate things. If it's a slow onset drug, as Nora was saying, it tends to be cause less different changes than a fast onset drug. And there's lots of data there with CFOS studies and ARC studies showing very different effects of fast-acting drugs and slow-acting drugs. So, you know, getting there slowly can be important there. But, and here again, and there, I think we can think about agonist-induced signaling here as well as a potential modification of drugs to affect their addiction potential. So you take a drug again, 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 you get all these adaptations which occur. You get changes from the goal-directed circuitry to the habit circuitry. I'm sure somebody talked about that, right? Yes, many times. Okay, so it's kind of old hat, no point talking about it. And all these adaptations which we know occur at the molecular, cellular, and behavioral levels. We lose euphoria from the drugs that we're taking, and also lose interest in natural rewards. The system becomes changed in many different ways, becomes insensitive. We get this loss of prefrontal cortical control, which is sort of what was that? Did you see that? Yeah, weird. Anyway, I thought I was seeing things. I had too much wine for lunch. Well, I only had a little bit, because otherwise you'd be hearing some rubbish. Anyway, so you might be hearing it anyway. So this prefrontal cortical control, which again is belabored in the course, I hope, by lots of people. I think Nora talked about it as well, but lots of people have talked about it. And after you stop the drug, obviously there's the withdrawal, all the opposite effects of the acute drug, and this dysphoric state, which Charlie was talking about a lot as well. You take your drugs again and again and again, you get this dysphoric state when the drug's gone, and as most addicts say, they're just taking their drugs just to feel normal again. And so you've had all these adaptations which have occurred. Again, these are going to be different depending on the drug which is hitting the receptor, and potentially important. And then you have this prolonged abstinence where cues, mood, stress, periods maybe, and certain genetics can trigger relapse. Again, if you can control some of these, especially the cues, the mood, and maybe the stress, these endophenotypes which can make you susceptible to relapse, perhaps we have other drug targets there. So the bottom line is there's lots of places within this cycle you can modulate addiction. And I want to talk about a couple of these, but before I do, have you seen this before? This darn opiate system. I mean, it's pretty amazing, this system, because it has, it's clearly evolved from one system. I mean, these are gene-duplicated receptors. They have intron exons in the same places. They're highly homologous. But what's happening with the kappa receptor? You activate the kappa receptor, it's aversive, right? You activate the mu receptor, it's rewarding. And I'm very interested in evolution, and I'll talk a little bit about that in a second. But how did this system get put together? How did it get put together? The same genes, the peptides are very similar as well. They all have, we won't talk about that in a second. But they all have this Tigagliphi med or lu at the end terminus. And then they have this C-terminal sequence which directs it to different opiate receptors, or have differential affinity for different opiate receptors. These are two of my favorites, dynorphin and BAM18. And then this other opiate receptor, which actually that system was not known until we did the cloning in 92. And this is a very much more abundant receptor. This receptor for orphanin FQ, we call it NOP, and there's many more receptors in the brain for NOP than there are COP, DOP, and MOP, which is the kappa system, the delta system, and the mu system. So, as I said, these receptors all clearly belong to the same family, same intron exon boundaries, very highly homologous receptors, clearly through evolution. Came from the same evolutionary start point. The as Charles informed you, these are protein precursors which are clipped in specific places to give you nearly 30 different opioid peptides. So there's like 30 of these different endogenous opioid peptides hanging around the brain. And they all have different affinities for the different receptors. Now, as I said, I'm pretty interested in evolution. And I've gone back in time to try and figure out how this system got put together, right? Because I want to know how does any system get put together? How do you get a neuropeptide precursor and a receptor linked to certain behaviors? And in this case, we have systems linked to completely opposite behaviors, some rewarding activities and some dysphoric systems. And maybe these systems actually are very closely linked. They're giving us information on how we decide about our environment. Are we going to be is this stimulus going to be rewarding or dysphoric? I mean, maybe this is how the system got put together, but I'm interested in that. So I've gone back and I can't find anything in the invertebrate system. So this is an interesting system. It just started in the vertebrate system. So those of you working on flies, sorry, you can't work on the opiate system unless you stick an opiate receptor in flies, which maybe is a good idea. I don't know. But the lowest species, unfortunately, for me, is this hagfish. You know what hagfish are? Slime fish. Have you seen these? You know, they're full of slime. They're horrible things to work with. But anyway, I've cloned all the receptors from these, and there's 3 opiate receptors and 3 or 4 opiate precursors in these, and they're almost identical to the precursors in to human, except they're a mix and match of the mu delta and kappa and ORL1 receptors. So this system is suddenly there in the hagfish, nothing below, and now I'm trying to find the genetic context of these receptors and ligands to figure out if I can find anything in invertebrates or somewhere that would explain how this suddenly got put together. Well, there's 3. So anybody got any ideas how a system can get put together like that? No?
Related transmitter system?
Somatostatin receptors are the closest. And if you go down to sea squirts and lancelets, you find somatostatin receptors. I mean, I've gotten lots of ideas, like I wonder if the receptors came first and they were chemotactic for food or something, and maybe they laid their eggs in, I don't know, the mussels we ate for dinner, and maybe there was some sort of transvection thing which happened. But I'm interested in how these things got put together. And that's my research. I don't let anybody else in the lab do this. Yeah, Scott.
It seems like you're focusing on the different behavioral qualities of, say, capital-good versus molecular receptors. But it seems that isn't that more of a function of the circuits that they're expressed in, or is there?
Well, I'm not even sure that they're in the brain. When I've done the analysis, they seem to be in the immune system, or they're in some kind of organs which are not part of the brain.
There's some GI-coupled modulatory pathway that's especially important to us because of the circuits.
Well, yeah, it is. It's important, but my question is, how did it get put together right in the beginning? There has to be something which engaged that circuitry in food reward or something, and that's what I'm interested in. How did that get put together? As I say, it's my project. I wouldn't let any of the students in my lab do this. This is my screw ups, but I'm interested in it. And as I say, I mean, the good thing is I've found like 5 or 6 new opioid peptides because of it. And, you know, evolutionary studies can be very enlightening, actually. You shouldn't just keep to the 1 or 2 species that you're used to working with. So as I say, I'm interested in that. Okay, so we got all this processing going on, and lots of different opiate peptides form from propimelanocortin, which also has the precursor for ACTH, which is involved in stress, right? It's involved in stress. All these systems seem to be involved in reward and stress. Proencaphalin has multiple copies of the encaphalin sequence. Prodynorphin, which Charlie talked about this morning, has these three copies, and they're all leuencaphalin. All the ones in the hagfish are all metencaphalin. There's no leuencaphalin in the hagfish, which is sort of interesting. And then there's often in FQ, which only has one copy. And depending on the processing, you can get activity at all these different receptors. Here's all the receptors and all the affinities for different the mu, delta, and kappa. And a couple of things you can see is that the proencaphalin here gives sort of delta and mu preferring. The prodynorphin gives mostly kappa preferring, and the POMC gives you mu delta preferring, as rather like the proencaphalin. In most instances that have been looked at, the knockout for the mu receptor is phenotypically more or less identical to the proencaphalin knockout in most circumstances. And the prodynorphin knockout is, the phenotype is more or less similar to the kappa knockout. So that's how we kind of know the pairing up. We don't know otherwise because these ligands are all over the place. They're expressed not coherently with the receptors. It's kind of like there's a mix and match, and you just see what works, which is kind of interesting. Yeah? Huh?
You just gave me my answer.
Yeah. There's no. It's a mix and match, and Charlie said that these can, work at distances away from where they're released, and so sort of give a sort of neurohormonal type tone to circuitry, and this is how we think they work. I want to go back because I wanted to tell you that there are these endomorphins, and there has been talk of endogenous morphine, where there is no precursor for endomorphin which has been found yet. This is a small 4-amino acid peptide, and everybody in the field is a little bit worried whether it's existing or not. So that is a problem. And then there's been talk over the years of endogenous morphine. It's potentially possible, but a lot of issues have come out with the food that people are eating. That it gets metabolized, and there's some issues there. There are some papers which look pretty convincing where they've controlled all the diet and everything, but it's still a bit of a question, certainly a question whether there's endogenous morphine in humans. The drugs that you all know about work primarily at the mu receptor, but as you heard, there's some different effects, such as buprenorphine, which sees the kappa receptor as well, and I'll talk a little bit about buprenorphine. Okay. So look at diversity. And I think this is very important. I mean, I know this is kind of silly in some ways, but we have 4 different genes, and we have alternative splicing, and some of these seem to be very, very rare alternative splicing, and some of them seem to be very common. But you can say, I guess on the average, there's five forms, different forms per receptor. Probably one of the biggest areas of diversity is different translational modifications. So, I'm guessing around 10 per receptor, including differences in glycosylation, palmitylation, Probably the biggest diversity is in the phosphorylation codes. These are very, very different, and they're agonist dependent sometimes. Sometimes these are just endogenously found, which they're phosphorylated. But agonists can change this phosphorylation barcode quite readily. Well, one of the biggest problems has been this monomeric, homodimeric forms. And so now we've got these receptors which can form heterodimers. And the evidence is getting more and more convincing that this is happening, in my view. It's been a very controversial area, but I think it's pretty clear now that heterodimers exist. How important they are in the action of these drugs, we're still not quite sure about. I'm going to show you one example of the alpha-2A and the mu receptor, which seem to be, traffic together when you put on agonists. And then probably the biggest diversity is the different protein associations. So the different G proteins which combine to receptors, the arrestins, kinases, phosphatases, and you're guessing about 50 of those. So if you calculate this out, we're looking at like 10,000 receptors, right? All with slightly different features, which is kind of scary. It's kind of scary for us pharmacologists because the potential is for every ligand we use to slightly differentiate between these receptor complexes. And you shouldn't be scared of it. You should just be aware that there is the potential for different activation of these receptors. So, and I know Charlie will have great sympathy when I say when Joe Blow says they can't repeat your results in a different system or even the same system in different receptor conditions, receptor heterogeneity may be the explanation. You can't say just because you're using a different system with the same receptor that that if you get one result and somebody else gets a different result that they're wrong. Receptors are very environment dependent and history dependent. So we look at the crystal structure. Well, this is just one complex which has been crystallized at one point in time. But obviously it's giving some lovely information about the structure the receptor can form. And, probably there's twists and turns, whatever you do with the different proteins binding, et cetera, et cetera. But this gives us a good base for figuring out how the ligands may interact with the receptor. So it's straight. Now, you know all this, that mu agonists are involved in analgesia reward. They stop your coughing, respiratory depression, constipation and nausea. The antagonists, at least in rats and mice, are very aversive. If you give naloxone or naltrexone to a mouse, they hate it. In fact, it's almost a stronger aversion than morphine is a rewarding agent, which is quite surprising. Now, we give naltrexone to people, and it doesn't seem to be so aversive as it is in animals, and so there is big differences. The delta agonists, they're not rewarding, and as Brigitte said, they seem to be involved in an antidepressant and anxiolytic effects, and I'll show you a little bit of data on that. The kappa agonists, which you heard about all this morning, evolved in aversion and hallucinogenic. The salvia, I know nobody admitted to taking it this morning, but my graduate student did take it. And he had some wild stories to say. He felt like it was kind of a rebirth. It was one of the most dramatic experiences he's ever had. So it is a very potent hallucinogen, and it's still legal, at least in the States. I'm not sure what's the status is here, but it's get. Yeah, you can still get it. Still get salvia online, yeah. Yeah, people were passing it around my talks. Yeah, in my lecture when I was giving a lecture. That's crazy. Yeah, I know. No wonder they didn't do well. That's all I can say. Anyway, so and then the ORL1, which is our most abundant receptor, is kind of bizarre. I mean, it blocks opioid analgesia. It's maybe hyperalgesic in itself. The antagonists have very little effect. We don't know much about this receptor, actually. It's just coming online, and we're trying to figure out what the hell it's doing there. And as I say, it's very abundant, but much more abundant than the others, but we don't know much about it. And I think that's the same as many G-protein coupled receptors in the brain. We don't know what they do. We only know about the opiate system because morphine has such dramatic effects. The opium poppy has so much dramatic. That's the only reason we know anything about it. Otherwise, we'd be still flustering around. What's this receptor? What's its ligand? We wouldn't know. I guarantee it. It's just because of morphine that we know about these receptors. So all drugs are dirty. You know that. You keep hearing that, and it's very, very true. Buprenorphine is a good example. Even naloxone is a good example. Naloxone sees lots of different other proteins in the cell. It sees filament A, and it sees something in microglia, which stop it working, which stop microglia being activated. So these drugs are not simple. And whenever you give a drug, you have to consider all these things. Okay, so let's have a look at a little bit at buprenorphine. It's working well as for maintenance therapy. It's a high affinity mu partial agonist, and that's what people consider it's important for maintenance. ORL1, it's a very low affinity partial agonist. Delta, it's a high affinity antagonist, which may not be good, right? Because remember I told you about delta agonists being anxiolytic. So if you have an antagonist at the delta, and as Brigitte was showing, if she did the delta knockout, she had an anxious animal. So buprenorphine may be interesting if we could get away that delta activity. It may be a better drug. The kappa antagonist activity, actually Mary Jean Creek thinks it's a weak partial agonist because she gets prolactin release, so she thinks it's a very weak partial agonist. But it's a complicated drug, and there's probably room for improvement. And so it's not simple. And people are now trying to combine this drug with naltrexone, which is a pretty good mu and delta antagonist, to try and develop what Charlie was talking about, have a kappa antagonist drug with all the drugs which are already accepted. So that's ongoing work, and I'm not sure there's some clinical trials going on there. Do you know what the results are of those? Well, it's too early. Yeah. They're still testing safety of that.
But for naltrexone buprenorphine, it seems to be promising in an open label treatment-resistant depression.
Okay.
So we'll see what happens.
So this is, you have to use combinations of what you got already. So this is in trials, but we'll see how it goes. Yeah, and this is some very recent data from the lab where we show we can block the effects of a kappa agonist on analgesia with a delta agonist. So here we've this is naive animals, and in naive animals, delta agonists are not analgesic. That changes in neuropathic pain, but we'll talk about that a bit later. But kappa is a pretty good agonist, shows an analgesic response. But we can block that with a delta agonist. So here again, kappa is being opposed. Maybe the anxiogenic properties of the kappa agonist are being partially obfuscated by the anxiolytic properties of the delta agonist. So we're losing the analgesic efficacy of kappa by putting a delta agonist there. Two agonists and having opposite effects. We're able to block the analgesic effect. And we can actually also block, in this paradigm, we can block the kappa analgesia with diazepam, which is a benzodiazepine. So we think that at least part of the kappa analgesia is a function of a sort of stress-induced analgesia. You know, it's dysphoric, right? Dysphoria makes you stress. Stress gives you analgesia, stress-induced analgesia. So some of the analgesic component of kappa, we think, could be due to the anxiogenic effects of.
Do you remember the dose of diazepam for reversing the?
I think it was like small, 10 mg per kg. But it reverses it. But you would have thought benzo was analgesic.
Right.
It's reversing the analgesia of a kappa agonist.
Mm-hmm.
Huh?
10 is very high.
Is it 10?
Very, very, very high.
Well, I thought it was 10 mg per kg. I'll have to look at the paper. I'll look at the paper. I wasn't quite sure. Wasn't quite sure. Can't remember.
So, Chris, one of the amazing things about the new agonists, of course, are the fact that they hit the pain circuits in so many different ways.
Right. Yeah.
When you dissect pain, it's not just tail pain. It's not just all.
No. There's lots of sites, and we'll talk about this. There's lots of sites where mu agonists can work. They can work in the skin. They can work in the dorsal root ganglia. And I've got some work I'll talk about the dorsal root ganglia. They can work in the periaqueductal gray in the brainstem. They can work at the cingulate nucleus, and you saw from John Carr's work, they can work in the reward system to be analgesic. Lots of different places. And we think the same thing happens with reward, that there's multiple sites in the brain which can trigger mu opioid reward. And in fact, there's some opiate reward which is dopamine dependent and some which is dopamine independent. And the circuitry of that is not well understood at all. And that's part of Brigitte and my work. We try to work this out with the knockout mice, and Raphael's doing some of this as well. Work out where and how the different reward circuits are modulated by mu agonists. So I think it's important. I'm going to go back to a long time ago. This was actually, it was around 25, 26, I don't know, a long time. And actually it was around the late, it was probably 1989, somewhere around there. That's ancient history for you guys. I know some of you might not have been born. I don't know, but no, probably you're all born. Just. Anyway, I was working with Mark here, Mark von Zastro, and we're working with NG108 cells. This is the cell we eventually cloned the delta receptor, and Brigitte, we did the same cloning strategy, more or less. And we were working with a cell line. And actually, I was very excited about this because we could treat these NG108s with a drug, etorphine, which is a potent agonist at delta receptors. We could wash out the drug and harvest the cells. And then we could do diprenorphin binding. And in control cells, we got very nice displacement of our diprenorphin, which we use for binding, by, this is daddle, which is an opiate peptide. But after treatment with the etorphine, although the diprenorphin still bound very well, we couldn't displace it with the peptide. Okay? So what do you think was happening? I'll try Scott's approach here.
Change the conformation of this receptor?
That's what we thought. We thought we'd twigged the receptor in a different conformation, and now the alkaloid would bind and the peptide wouldn't, because we tried a lot of alkaloids to see what would displace, and we could only find alkaloids which displace the diprenorphin and not peptides. So we thought we'd had this amazing change in the receptor which twisted it around and made it into a receptor which bound opiate alkaloids but didn't bind opiate peptides. We were wrong. Any other explanations? Good idea. Non-specific binding has a possibility as well. Any, you should know. No? Yes, that's right. It was just a simple internalization of the receptor. So alkaloids get through membranes. They cross membranes very easily. The peptides don't in general. They only see surface receptors. So what had actually happened was the receptors had nipped inside, and they were still binding the alkaloid. So the alkaloids could all go in and displace the tritiated diprenorphin inside the cell, but the peptides were stuck on the outside of the cell because we were doing whole cell binding at this point. And so this got us really interested in trafficking of opiate receptors. And me and Mark went on to do a lot of work with the NG108 cell line. And one of the important things we showed was when we treated with morphine, although the signaling was still there, the morphine didn't produce this effect, but etorphine and daddle could make this occur. And now we know that morphine is a drug which is very, very poor at internalizing opiate receptors. Not only the delta receptor, which is the one which is in the NG108, but also the mu receptor. It's a very poor internalizer of receptors, opiate receptors. So what do you think that means? Do you think it means that opiate receptors, when they're hit with morphine, are going to desensitize less because they're still on the cell surface? Do you think so? You know, they're still there. They're not going to internalize, so maybe they're going to be resistant to desensitization. What do you know about morphine? It does cause desensitization, right? It does cause tolerance. It does cause the receptors not to work very well. It doesn't necessarily mean there's no tolerance. But and I'll show you some data coming up. Well, actually, Brigitte showed you some of that data. Even though the receptor didn't internalize, it still became unable to signal. Charlie's shown you some of exactly the same data where he treats with an antagonist, and it's still on the cell surface, presumably. I expect you know, but it's not signaling. So just because it's on the cell surface does not mean it can signal. It does not mean it's signaling. Anyway, we went on to do a bunch of studies here, and this is just showing you the transfected cells, because now we had we could epitope tag things, put little tags, and follow these receptors around the cell. So we did a whole bunch of this work, and this is just in 293 cells with transfected with the epitope tagged, flag tagged receptors. And this is actually mu cells, but you can see that etorphine causes this robust internalization, as Brigitte was showing you last with deltorphan and SNC80, right? You get this internalization with the delta. And Damgo also causes this internalization. Etorphine does, but it's blocked by the antagonist naloxone. And morphine, it still sits on the surface doing nothing, right? It's still there. Even though daddle and morphine have very, very similar signaling with regard to adenylate cyclase. The trafficking is very different. Complexes. They're creating different complexes. The daddle and the etorphine are causing GERK-mediated phosphorylation of the receptors. These receptors bind arrestin, and they go inside. Morphine does not cause nearly so much phosphorylation, creates a different complex, and it sits on the cell surface, but it can still desensitize. Okay, so there you go. So this is a review that Brigitte and myself wrote a few years now, and just to show you all of the things which can happen, we have this dimerization. Lots of kinases can interact with these intracellular domains, and obviously the G proteins. We have other proteins which can bind to the receptor itself, like the arrestins, calmodulin, and filamin, and these are involved in, all these are involved in this trafficking and the signaling of the receptors. These are the ones we know of. These are just the ones we know of. There's probably many others still to be found, but this is what we know of. So obviously the complex will depend upon the cellular proteome, whether they're expressing different arrestins or whether they're expressing calmodulin or filamin. The history of the
Is there any information on the stoichiometry of the receptor and the other attachment proteins? So
it's going to be different in every cell. I mean, you know, depending on where it's expressed, you're going to have the different associated proteins. So some cells, there may only be arrestin 1, and others there are arrestin 2. And there's going to be differential expression of all these signaling and scaffolding proteins and proteins which interact with the receptor. Clearly, there's different heterodimers formed in different cells.
oh, I mean the numbers. So one heterodimer, one G protein.
One heterodimer. I don't think anybody knows about the stoichiometry is going to be different, certainly in every cell. Yeah.
Differentiation in the associated protein is by cell or tissue by tissue?
Cell by cell. My guess is that every cell is kind of like a unique one. I mean, obviously, it's not totally unique, you know, but every cell is going to have a unique expression profile. So you're going to see differences in D1s and D2 cells, or interneurons is going to have different proteins, other G protein coupled receptors that they can heterodimerize with. It's the complexity is much greater, and you should be aware of it, but you shouldn't be frightened of it. I don't want to frighten you guys and say that it's so complex that you can't work on it, because that's not what I'm trying to get across here. I'm just trying to get across that this is a the opiate receptors are much richer than we thought, have a much richer pharmacology than we thought. You know, we used to just think of methadone, buprenorphine, morphine. The richness is, and the potential for finding compounds which are specific is amazing. Yeah?
What are the differences in the subcellular targeting of the different complexes that they form? So are there complexes that get formed
Okay.
like in the dendrites and then cell body?
That's another possibility, that there's different compartmentalization of these different complexing proteins, so that receptors may be behaving differently in the cell bodies than in the end of the processes.
I couldn't tell from the data yesterday whether the endocytosis rates are different.
It's a good question. Well, Mark von Zastra did an experiment where he found that in the processes, in the dendrites, that morphine caused internalization, but not in the cell bodies. And I mean, if you increase the concentrations of beta-arrestin 2 and GERK2 in the cells, you can get morphine to internalize. So you can play around with the stoichiometry or the likelihood that the receptor's going in. I mean, all of these are sort of slightly different. You can sort of force things to go one way or the other depending on the expression. Yeah, Scott.
Richness of control that you pointed out for opioid receptors, is this kind of a model for, in your sense, for many other GPCRs that perhaps less extensively analyzed? And so, you sort of,
Yes.
if people are going to be afraid of this, would you just expect that if they studied any other GPCR, they'd find the same thing? It may just not yet.
Well, I think we're so fortunate because we have a richness of the drugs. And most areas you don't have this richness yet of ligands which interact with the receptor. And this sort of, the beta-adrenergic receptor is another one which shows these kind of effects. And bronchobilca is very
General regulatory processes that are just worked out in the system. Is that kind of the right take home message?
well, the take home that I have is that, well, we actually don't know about the endogenous opioid peptides. We haven't done the agonist-directed signaling of those, and this is probably my fault. I probably should have done it at some point, but I haven't. I know there are some people interested in doing this now because they have all the different assays set up for all these different signaling cascades. But I think what we'll find is that the endogenous opioid peptides are all going to have different sort of spectrum of signaling cascades. So depending on the processing of the peptides, we're going to amplify certain cascades and not others. And I think what happens is that we just happen to have all these drugs which stabilize different conformations of the receptor. So we're able to see this in the opiate system, and it just hasn't. We just don't have the drugs in other systems to be able to see these things. But I think it will happen. I think you'll see this with almost every receptor, that you're going to stabilize different conformations. You're going to get different signaling cascades happen. Yeah.
And this is just a curiosity. I mean, I can understand the
Lipophilicity?
no.
Sorry,
I can understand.
I don't understand your accent.
I can understand the advantage of heterodimers.
The heterodimers.
Yes, I can understand that could be an advantage to form heterodimers. But what about homodimers? What is the advantage?
What is the advantage of forming a homodimer? The question is, can you actually? A lot of people have actually questioned whether you can actually have a monomer. I know you can crystallize it as a sort of monomeric form, but a lot of people question whether the active form is not the dimer. And if you prevent dimerization, usually the receptors don't work. Anything you do to muck up dimerization seems to inactivate the receptors. So they probably are working as dimers, but it's so difficult to prove. Everything you do to try and prove it, you can think of an argument against it. That's the problem. It's been very difficult for people to prove whether you're forming dimers or heterodimers or whatever, or whether they're working as monomers. I mean, some of the evidence, some of the really good evidence is you can do point mutations in receptors which inactivate them. But when you make two different ones, you make two different point mutations, you have two inactive receptors when you express them, but when you express them together, you get an active receptor. That's the best evidence that I've seen that you form heterodimers. And then there's some trafficking evidence, and I'll show you some of the trafficking evidence in DRG cells, which suggests that they're acting as dimers, and then we get agonist-directed signaling for the formation of the dimers. But it's complicated to prove. It really is. It's not easy, and it's still a question in the field. And the cannabinoid receptors are another mess, right?
So the presence of delta receptors have also been indicated in tolerance for morphine.
They have.
Can you speculate on the mechanism? Is that possibly due to arrestin-mediated jump activation?
Well, there's some controversy on whether delta receptors are really, really heavily involved in and tolerance. I mean, you still get pretty good tolerance in the delta knockouts, and Brigitte's done this. You get a little change, right?
Yeah.
You get a little change. It's difficult because if you take away the delta receptor, you have a more anxious animal. So it's maybe the anxiety is affecting the tolerance paradigm. Lots of things affect tolerance. There's many ways of affecting tolerance. Stick in many kinase inhibitors and do lots of things to interrupt tolerance. So my guess is that this may actually be a behavioral thing. There's no proof to me that it's a heterodimer regulated event. Although there's pretty good evidence that you can form heterodimers in vitro, the in vivo data is pretty poor from what I can understand. There's not really good evidence for it. Okay? So, okay, so, I just said all this. No point in saying it again. Okay, so these are these are the receptors which have been implicated for other G protein coupled receptors where you can form heterodimers. Mu-D2, I don't think mu is expressed in D2 cells, right? Not very often. I guess it can be. Yes, it can be. It can be. It's definitely expressed in D1, but it's much less expressed in D2. So you can form the somatostatin receptors, which are very similar to the opiate receptors. The mu and the cannabinoid receptor has been postulated, shown. All of these, there's some evidence that mu receptors and delta receptors can form heterodimers. I'm not sure if there's any evidence. Oh, yes, there is some evidence for kappa as well. Yeah, the beta-2 adrenergic receptor. So there is some evidence for heterodimers from all these. But the proof, as I said, is very difficult. You know, you can do the pull downs, but usually this is done in overexpressing cells. The expression of the two inactive forms is my favorite. This happens, you know, GABA B doesn't work unless it's a dimer. The GABA B receptor has to be a dimer, otherwise it doesn't work. So that's one example, clear example, where you need to have a heterodimer. There's lots of evidence that these can. Phil Portuguese is doing some very interesting work where he takes a ligand for the opiate receptor, and then he puts a different linker on, and then he puts another ligand for a different receptor on. Okay, so he's made all of these different compounds with different lengths of the linker, and he finds this incredible change in activity. I think one of them was 3 million times different in activity, just depending on how different the linker was together. So again, this is giving a more sort of credibility. The credibility for dimers is growing very strong. That's all I can say, in my mind.
Don't you think that these systems, these receptors have evolved not for Phil Portuguese's compounds, but for the endogenous system? And
Yeah.
Going back to the question you raised earlier, which is if these heterodimers are really going to be functional, really important, it has to do with the way in which the enkephalin, proenkephalin products are cleaved and interact differently. So you got to go back to the lab and really tease apart the enkephalin analogs and look at how their physiology changes depending upon which combinations.
I don't think we know which we don't know the heterodimers, which may be relevant yet. That's going to be another whole area, I'm sure. Yeah?
Excuse me, sir. Is there any data about that?
Don't call me sir, please. Please. It makes me feel old and like a school teacher. Okay, yeah?
Is there any data about interaction with the NMDA and the opiate receptor?
Yes, there is. But I feel that's probably more likely to be signaling and phosphorylation, making these big complexes where you get signaling complexes, and you can, you recruit kinases, et cetera, and then they phosphorylate and change the NMDA receptors. So there's lots of ways of things working.
Because yesterday, as also you have it present here, I mentioned that quinolinic acid, and it's its structure analog, this KINO1, which we examined in the opiate receptor.
Right.
They don't have any direct binding to the opiate receptors, but at the same time we can see some changes in the G protein activity, which is in the presence of the ligand, of opiate ligand, we cannot get this proper activity, and it's decreasing in the activity. Can its effect in the somehow NMDA receptor or other receptors can affect them?
Yeah, I mean, that's what we're saying, that the NMDA receptor can be part of the signaling complex, and actually dynorphin itself will interact with NMDA receptors. There's a long history of this. So some of the opioid peptides directly interact with NMDA receptors. And NMDA receptor blockade can block tolerance. So, I mean, the system, you've got to think of all these complexes which could form, but please don't be scared of them.
No.
Please don't be scared of them. They're just... Otherwise, you couldn't work. That's right. I mean, you've just got to see it as a rich area, and you've got to see the possibilities of that as we evolve in our knowledge, that we'll be able to get some more specific ligands which may differentiate different behaviors. So just to be aware that a receptor can be many different things. And we had this discussion actually at INRC this year. What is an opiate receptor? It's are there 10,000 of them, or do you define it by an antagonist activity? How do you define it? Or the knockout? Do we define it as the knockout? The lack of activity in the knockout. It's an interesting question that we're kind of all grappling with. Anyway, okay, so let me just show you some of our data. This is our antibody, which actually is quite nice. This is our this was our antibody, which we now have nothing of, so I don't know why I'm showing it to everyone. But you can see really nice distribution it kind of looks very much like Brigitte's pictures of her the red mice, the mutuary mice. We'll have to look and see if there's any cell bodies there. Anyway, there's lots of different areas. So these stars are where you can micro-inject opioid peptides and get relief of pain. So lots of different areas that you can get relief of pain. Again, it's the system seems to have evolved so that there's many different areas where you can block the pain pathway. And as I say, I think we're going to have the same story with the reward, that there's going to be lots of different areas where we can get reward with opiate drugs. So let me just quickly go through this story. This is a story of alpha-2A and mu receptors, which are co-expressed in these DRG cells. And basically, what we can do is we can treat with Damgo, and this is the normal clonidine inhibition of, this is the alpha-2 agonist. This is the inhibition of calcium currents with the alpha-2A agonist clonidine. If we treat with Damgo, we basically lose this activity, and this can be blocked by CTAP. So CTAP's a mu antagonist, and it blocks the Damgo-induced effect. We also see that if we treat with Damgo, then we get co-internalization of mu in the alpha-2A receptor. So, it's some evidence that maybe mu and the alpha-2A are forming heterodimers, right? Any other explanations? Are there any other explanations? It could be a signaling cascade, right? It could be that you're activating maybe MAP kinase and MAP kinase, or one of the G protein receptor kinases, which somehow causes phosphorylation of the alpha-2A receptor, which then initiates a cascade where the receptor goes inside the cell, right? So there are other explanations besides it forming heterodimers, that's all.
But normally, G protein coupled receptors are not phosphorylated without ligand, yeah? They need the ligand to be phosphorylated.
That's usually true. That's usually true, but there's some endogenous phosphorylation as well. So you can activate, you can activate PKC or pKa in the cell, and you can get some other phosphorylations of these receptors. And so that some of the receptors at resting state have a certain barcode, or what Bob Lefkowitz calls his barcode, right, which is a different phosphorylation signature. And that changes, yes, with agonists, and you tend to get more phosphorylation. And depending on the agonist, you get different barcodes formed. And that's probably very important for the complex formation, which happens with each agonist. Let me get on and show you what happens when we treat it with clonidine. So if we clonidine pre-treat, we get exactly the same thing. When we treat with Damgo and even morphine, we get this a desensitization of the mu receptor. So if we treat with clonidine, we also get the mu and the alpha-2A going inside the cell. So we seem to get co-trafficking depending on the agonist. So what do you think happens with morphine? Morphine is going to activate the mu receptor. So if it's a signaling cascade, you might expect morphine to cause the internalization as well of the alpha-2A. It's activating the receptor, but it doesn't cause any desensitization, or it doesn't cause any internalization of the alpha-2A or the mu opiate receptor. So it only happens with ligands which cause internalization of the mu opiate receptor or the alpha-2A receptor. Then we tried to find things which blocked this desensitization. And what we did is we found that Damgo, but not morphine, could activate P38. This is with a flow cytometry assay that we developed. And we found, because of this, we tried the P38 inhibitors. And we found that we could block the Damgo-induced internalization of mu receptors, but we also blocked the cross-desensitization of mu and alpha receptors if we treated with Damgo, but clonidine still induced this desensitization of the receptors. So clonidine is still causing these changes in the receptors, even though morphine is now still stuck on the receptor, stuck on the cell surface. Okay? So here we started differentiating them, and then we also used in the beta-arrestin knockout mice, we showed that actually clonidine, if we treat with clonidine, we actually lost the internalization of the alpha-2A, but morphine still actually goes into the cell, so we could differentiate the two. So they're not always together, right? You can separate the two receptors out, but when you activate them with ligands which produce internalization, they go in together. Now, my explanation or my hypothesis is that the ligands themselves are initiating the dimerization. And that's where I'm at this, and it's difficult to prove, obviously, but that's where I'm at on this. And we've done quite a lot of work with the arrestin knockouts, and these are the conclusions that we have, that with a morphine-induced complex, we're involving arrestin-2, involving JNK, many different kinases. But it still causes desensitization, but it sits on the cell surface. When we have a Damgo-induced complex, we're activating the GERCs, GERC2, and there's arrestin-2 is involved, but P38 is activated, and this induces a complex which causes the internalization of the receptor. We've also shown that there's a constitutively active complex, which, if we take away arrestin-2 from the cell, we get a constitutive activity of the mu-opioid receptor. So that's another thing we haven't talked about, right? Constitutive activity. I haven't heard constitutive activity being, but some of these receptors can get into a mode where they're constitutively active. They don't need a ligand to work. They just work by themselves. And the cannabinoid receptor is a very good example of a constitutively active receptor. So is the delta, which is one of the first ones to be found. Okay, so this is work. Work from Amina, who used to be in Brigitte's lab, and she came over to my lab for a postdoc, and she's got some very interesting data. Here she's looking at the arrestin-1 and the arrestin-2 knockouts. So most people will tell you that arrestin-1 and arrestin-2 probably do the same thing, right? Yeah, no, you probably arrestin. There's many different arrestins. There's actually 3, and depending on how they're called, you know, they've been renamed a few times. But I'm going to call these arrestin-1 and arrestin-2. This is the old nomenclature. Now it's 2 and 3, but they don't necessarily do the same thing. And in fact, what Mina showed here is that arrestin-2 is actually protective against desensitization from arrestin-1. That's a very interesting story. What she's done is here, if you treat animals with CFA, which is an inflammatory into the pore, you upregulate the activity of delta receptors. And if you look in the arrestin-2 knockouts, this is the wild type. If you look in the wild type, and this is an injection 1. I think this is what Brigitte basically showed you yesterday. With SNC80, you get a nice activity, but if you inject 4 hours later, you get a very reduced activity of the analgesic activity of the SNC80. ARM390 after 4 hours shows very little desensitization or tolerance, whatever you want to call it. It still works the same way. Now, if you take away arrestin-2, that's what she's done here, and you inject with SNC80, you still get the same desensitization. You still get the desensitization of SNC80, but you now get desensitization of the ARM390. So arrestins are supposed to be involved in desensitization, right? They're supposed to stop the receptor working. Not here. Arrestin-2 is actually preventing the desensitization of an agonist, which doesn't internalize. If you remember, ARM390 doesn't internalize a receptor, and the SNC80 internalizes it very well. So in the case of arrestin-2 knockouts, we start getting ARM3 desensitization after 4 hours. In arrestin-1 knockout, we have no change in the ARM390 because it's reliant on arrestin-2, and now the SNC80 doesn't internalize and doesn't desensitize. So SNC80 actually lasts for a long, long time if you take the arrestin-2 away. So anyway, so arrestins are not all the same, and I think that's an interesting story which we're trying to put together now with Amina. Okay, so out of all the studies which have come out from this agonist trafficking, it turns out that there are now some ligands. Most of this early work was done by Laura Bon, and she found that without the arrestin-2, there was some slightly, actually markedly changed properties of morphine. And now what they've identified is a ligand for the mu-opioid receptor which does not recruit arrestin-2 at all. It's called Trevena TRV130. And they now have this in clinical trials. And at least in mice, it looks very promising that it'll be better than the morphine with regard to tolerance and these kind of actions. So something came out of this, by understanding by screening for drugs which don't recruit arrestins. Okay, so I want to talk about some other sites of potential drug development, and this involves hyperalgesia. Now, this is kind of an interesting test. It's done in humans, right? And it's called the cold pressor test. What you do is you have a bucket of water, and you've got to put your hand in the bucket of water, and you say when you feel pain, and you say when you can't hold it under the bucket of water, of ice water anymore, so you have to take it out, right? And these are the two criteria. So this is the pain detection and the pain tolerance. This is work I got from Walter Ling, who's a clinical guy. And this is in methadone-maintained patients. This is 24 hours after their last dose. And you can see that the pain detection in the maintained patients is way down from the controls, okay? And the green is the controls here. And so the controls can leave, in this case this is pain tolerance, the controls can leave their arm in the cold water for like 60 seconds, right? And the methadone-maintained patients can only leave it in for about 20 seconds.
They do have methadone still. They have
this is 24 hours. This is 24 hours after the last dose.
After the last dose.
Okay. Now, this is 3 hours after the methadone dose, which is the optimum for most analgesic things. So you would think they'll be nicely analgesic, right? No. The whole system's desensitized. But not only is the whole system desensitized, it's showing hyperalgesia. You've got this hyperalgesic effect. So these guys are incredibly, they're still actually a little bit more pain sensitive in the pain detection, but they're still way below the controls, even though they're on methadone. There's no methadone in the controls. This is...
question, how about the methadone?
Well, if you had methadone here, they'd stay in forever. It's just that these are methadone maintained, so they have a completely dysfunctional opiate system. But not only do they have a dysfunctional opiate system, they're more sensitive in the control situation than wild types. You can't, it's very difficult to treat. You probably know this from the addicts, that it's very difficult to treat them for pain. These methadone addicts, you don't know what to do with them when they have a problem, which they need pain medications for. Very big problem. They don't know how to deal with this. So this hyperalgesia is something that is interesting to a lot of us. And there's some recent work which was published looking at the etiology of this hyperalgesia, how it may function, and some pretty elegant work which was published in Nature Neuroscience, showing that at least a part of it is mediated by activated microglia. Okay, Now, all of the presentations you've had thus far have probably focused on neurons, right? We haven't talked about the potential. I mean, Charlie mentioned glia just for a second, but glia are probably involved in a lot of the functions of how the brain circuitry gets organized. And in this case, there's clear demonstration that the microglia release BDNF, which then downregulates this chloride transporter KCC2. You probably heard about this, that GABA is excitatory during development. Right? Have you heard of about this? Well, the reason is that KCC2 is downregulated during development. Now, KCC2 is a chloride transporter. Okay? So it's throwing chloride. It transports chloride so that there's a good chloride gradient across the cell. So when you open GABA receptors, chloride rushes in. And then you get hyperpolarization. If you haven't got a good gradient of chloride, it can actually be excitatory. And that's what happens during development. So if you downregulate KCC2, you're actually causing this GABA receptor to be in the GABA cells, in the GABA receptor cells. You're causing a depolarization. Okay, so this is one of the mechanisms that they think could maybe lead to hyperalgesia, and they did some very elegant studies showing this dysfunction of the KCC2 after chronic morphine treatment, and how this is regulated by BDNF. We've actually been looking at this in the VTA, and I wanted to show you some quick data here. This is looking at activation of microglia in the VTA with a chronic morphine paradigm. And there's a number of antagonists. Minocycline is one, and plus naloxone is another, which is used to block this microglia activation. So does this have any functional consequence? Maybe, maybe not. Let's have a look. Okay, so we look at cocaine, the reward. This is a simple CPP, and this is opiate-naive animals. There's no effect of minocycline or plus naloxone. But in opiate-dependent, we actually lose the reward for cocaine CPP, rewarding effects of CPP. We've looked at dopamine release, and the dopamine release is almost zilch after chronic opiate treatment. It goes. We talked about this before, that if we...
When you say dependent, what do you mean? Are they in a withdrawal state or just chronic?
No, they're in withdrawal state.
How did you induce the withdrawal? Spontaneous?
Well, no, spontaneous, yeah. So they have morphine for, I think this is 5 days, an escalating dose. Mm-hmm. And then we do the dialysis, or we do the CPP is done. No, actually, CPP is done with this is cocaine, right? So this is you do the dependence first, and then you do the CPP afterwards. So yes, they're in withdrawal, yeah. So anyway, we think that microglia may be involved here. I mean, minocycline, again, is a dirty drug, so I'm not going to... It's a phosphodiesterase inhibitor. Plus naloxone, we don't know how it works. We just know it blocks the effects of microglia activation. So they don't work. But this is pretty robust. It's a very robust finding. And so I think that microglia should not be ignored. I think their activation does occur with lots of drugs and with pain. You get activation of microglia. They may be changing the circuitry, and we have evidence now that in the VTA we have dysfunctional KCC2, a downregulation of KCC2 activity with this assay that Anna's been using here. So don't disregard other cell types. Yeah?
So by inactivating microglia, you mean like everything also with make release and the consequent release?
Well, no, it's the activation of the microglia. It's not they change their form. There's an activated state and an inactive state, and this is just the microglia. It's not all the glial cells. It's just microglia. No, we've gone through lots of brain regions, and the VTA seems to be one area where it's particularly pronounced, this activation. We've actually gone through the whole brain. She's gone through the whole brain now, and this is pretty recent stuff. So, but she doesn't find it everywhere. It's not everywhere.
Do they block morphine dependence, these microglia inhibitors like minocycline and...
Well, it blocks the hyperalgesia. It doesn't block the tolerance.
How about withdrawal?
I don't know about withdrawal. There is some data from Linda Watkins, I think, affecting withdrawal.
That's why they're reestablishing cocaine preference, because...
They certainly are sort of normalizing they're normalizing the function of the reward system. So we're really interested in this. We've got a lot more work to do on this, but this is a bit of a teaser, but because we haven't worked all of this stuff out. But I mean, it's exciting stuff.